This is a production of Cornell University.
Okay, so cucumber, or Cucumis sativus, and melon, Cucumis melo, belong to the Cucurbitaceae family. And I know that this group knows that the Cucurbitaceae family includes many high-value and nutritionally important crops: cucumbers, melons, watermelons, summer and winter squashes, pumpkins, and gourds. And while it doesn't have all the wonderful genomic tools that Arabidopsis has, cucurbits are beautiful, and they taste great, and they can be really bizarre. Cucurbits exhibit tremendous developmental plasticity in fruit size and shape and pattern, and they're also extremely sexually flexible. So our primary interests are sex expression during floral development and fruit development, especially early fruit development, growth and expansion phases. So cucurbit species and genotypes within species exhibit a wide array of heritable sex types with varying combinations of either male or female or bisexual flowers. These underlying sex types can then be modified either by external environmental factors or the application of hormones. So if we think about sex expression at any given node along the vine of a cucurbit plant, that's going to result from the underlying genotype of the plant, the developmental stage of the plant, hormone balance, and environment. Now, cucurbits are typically either monoecious or andromonoecious. Monoecious means you have separate male and female flowers on the same plant. Andromonoecious would be you have male flowers and bisexual flowers on the same plant. In addition, there tends to be a developmental pattern for expression of these sex phenotypes. So if we're looking at cucumber, which is monoecious, you would begin with a series of vegetative nodes along the main stem, followed by nodes that produce only male flowers, followed by nodes that produce both male and female flowers. On the other hand, for the andromonoecious melon, analogously, you would have vegetative nodes, male nodes, and then nodes that produce combination of bisexual plus male nodes. So despite the fact that these plants can produce unisexual flowers, either only male or only female, at early stages of development, all four whorls typical of a bisexual flower will be initiated. So you'll have sepals and petals and stamen and carpal primordia all get initiated. Now this is from cucumber. If we look at male buds, we see the initiation of the stamen primordia and the very beginning of carpal primordia. But then as the flower develops or the bud develops, we see growth and differentiation of the stamen primordia, but the carpal primordia don't really do anything. In contrast, if we look at what will become a female bud, we see the stamen primordia looking similar to the male, and then the beginnings of the development of the carpal primordia. As that moves forward in time, the carpal primordia continues to develop. It develops ovaries and ovules, but the stamens are arrested in development. In addition, numerous studies with exogenous hormones have indicated that ethylene is the primary hormonal determinant of sex in these species. In species such as cucumber, melon, and squash, ethylene will promote femaleness, so if you spray a plant with ethylene, you'll get more carpal-bearing buds, while inhibiting either ethylene production or perception, for example, spraying with silver nitrate, you will increase maleness. Recently, key sex expression genes have been cloned both from melon and cucumber, and they have been shown to encode an ethylene production enzyme, ACS or ACC synthase, which is one of the key steps in ethylene production. So for example, in cucumber, female lines that produce only female flowers have an extra copy of this ACS gene. In melon, those lines that are capable of converting their bisexual flowers to become just female also express an additional ACS. So it has been proposed that sex determination in these developing floral buds results for a combination of ethylene production and ethylene perception by relevant floral primordia at critical times in development. So our questions were, can we identify critical sites for ethylene perception or production within the developing floral bud that are responsible for sex determination? And can we use that knowledge about the role of ethylene at critical developmental points to either increase or cause earlier carpel-bearing bud formation? And like was just said to you earlier, if you care about fruit production, of course, you care about female flowers or carpel-bearing flowers. So in order to ask, can we identify critical sites for ethylene production within the developing floral bud that are responsible for sex determination, we took advantage of a gene from Arabidopsis, the mutant ETR1-1 gene. This produces a protein that inhibits ethylene perception, and it is a dominant negative inhibitor of perception. And we introduced this gene under control of one of three promoters, either the standard cauliflower mosaic virus 35S promoter that drives expression, strong constitutive expression, the CRC or crab's claw promoter, which in Arabidopsis targets expression in developing carpels and nectary primordia, or the apetal 3 promoter, which in Arabidopsis drives expression in developing stamen and petal primordia. Now, we had predicted that if we inhibit, because we know if we spray silver nitrate and inhibit perception, we'll inhibit carpel-bearing bud formation. So we predicted that if we constitutively inhibit ethylene perception, that we would lose carpel-bearing flowers. We also predicted that if we inhibited perception in the carpel primordia, that we would inhibit development of carpel-bearing buds. But if that we inhibited perception in the stamen primordia, we would not have an effect on the floral development. So first then we did transformation of melon using agrobacterium-mediated transformation of cotyledon explants. We regenerated our plants, produced progeny, verified that we had introduced the genes with the appropriate promoters that we expected, and that they were being expressed as we expected. Looking first at the 35S or constitutive expression, we wanted to know that if we took that ethylene, the gene inhibiting ethylene perception in Arabidopsis, that it indeed would inhibit ethylene perception in melon. And we were able to see several phenotypes that confirmed this. For example, the transgenics were very inhibited in rooting, as people have observed with ethylene inhibition. They also showed a delay in abscission of the floral buds from the pedicels. And we also saw an increase in ethylene production. This also is standard for ETR1- mutants or other ethylene perception mutants. They're unable to perceive the ethylene. Normally, if they perceive the ethylene, there would be feedback inhibition in the level of ethylene produced. Now they don't know, so you see an increase in ethylene produced. So this convinced us that the gene, the ETR1 gene from Arabidopsis, was capable of inhibiting ethylene perception in melon plants. What effect did it have on sex? This graph shows nodes along the main stem. These are wild-type melons and also non-transgenic segregants. And from there we see initially vegetative nodes, followed by nodes that produce only male flowers, followed by nodes that produce either bisexual or male flowers, as we would expect. But when we look at the transgenic ETR1-1 melons, we see that the great majority, about 86% of them, never produced a carpel-bearing node. There were a few plants that produced one or two, and then returned only to producing male flowers. So this indicates that ethylene perception is indeed critical for the production of carpel-bearing buds. So the next question is, where in that floral primordia is perception essential? Now recall we had predicted that if we inhibit perception in the carpel primordia, we would inhibit carpel formation. But if we inhibited perception in the stamen primordia, we did not expect that that would have an effect. What we observed was the exact opposite. If we inhibited perception in the stamen primordia, we essentially eliminated any production of carpel-bearing buds. But if we inhibited perception in the carpel primordia, not only did we fail to inhibit carpel-bearing bud formation, we actually saw somewhat of an increase. And I'll come back to that in a moment. So after we were done scratching our heads, going back and retesting all the plants and making sure we hadn't confused which line was which and which had the right promoters, when we started to, the first conclusion had to be that ethylene perception by stamen or petal primordia is necessary for carpel development. Now this starts to make sense. The stamen primordia are initiated before the carpel primordia. So that means that they would be present to perceive ethylene in time to make a decision about whether or not to allow the carpels to develop. This is also consistent with the observation that inhibition of carpel formation in a male flower occurs at an earlier stage of development than does inhibition of stamens in what will become a female flower. So this also suggests then that there's crosstalk going on between these developing sex organs. Now I mentioned I would come back to why did we see increased carpel-bearing bud formation in the lines that had inhibition of perception in those in the carpel primordia. Well, if we look at the apices of those plants, what we see is an increase in ethylene production. Remember, I indicated that you inhibit perception, you can get an increase in ethylene production. So the carpels are not perceiving, we get an increase in production, but the stamens can still perceive. So they perceive the ethylene, that could lead to an increase in the carpel-bearing bud production. In addition, we saw a further phenotype, and that is, remember, typically we would have a bisexual flower, so you would have stamens as well as carpels present. But in the CRC ETR1-1 lines, approximately 50% of the buds and the flowers saw a complete loss of stamens. Essentially, they became female. So again, if you have this elevated ethylene, the plants go on to produce carpel-bearing buds, but then that elevated ethylene can subsequently lead to an inhibition of stamens. That's what happens naturally when you have plants that are capable of producing female flowers. You have that expression. In the case of melon, you would have the ACS7 gene that comes on later in development and causing the inhibition of the stamens. So collectively, these observations provide new insight into the sex determination process and interaction between the organ primordia. They suggest that the critical site for ethylene perception at the time of sex determination resides within the stamen or petal primordia, and conversely, that inhibition of stamen production results from ethylene production by the carpel primordia. This occurs naturally through the ACS7 gene in melon, and in our case, using CRC ETR1-1. Furthermore, this reciprocal relationship would then assure that at least one sex is expressed, i.e. that you don't inhibit your stamens until you know you've got carpels being produced. So we then want to look at what was happening with regard to ethylene production. So in this case, we used the ACS, or that first committed step in ethylene production, and hooked it up to the same set of promoters as I described earlier. If we look at it with 35S, we verified that there was production and that expression of the ACS gene was capable of driving increased ethylene production. That also led, as we would predict, to an increased number of bisexual buds and flowers and an earlier appearance of the first bisexual flower on the main stem. So this is consistent with the predicted effect of increased ethylene production. If we now targeted that ACS gene to stamen primordia, we saw a very dramatic effect on femaleness. Here again is the wild type showing vegetative nodes, followed by male nodes, followed by nodes that have a mixture of either bisexual or male nodes. But in those lines for which ethylene production was targeted to the stamen primordia, we saw a very dramatic change. We saw a complete loss of the male-only phase, and we saw the appearance of a bisexual-only phase that does not normally occur in wild-type plants. A subset of those individuals actually showed a complete conversion. They never produced male flowers, only produced bisexual flowers, essentially became hermaphrodite plants. Now when we look, we did see an increase in the number of carpel-bearing buds and carpel-bearing flowers, but even more dramatic than the increase in carpel-bearing flowers was actually a loss in the number of male buds and flowers. So that if we look at the total, we actually see total buds reduced in the transgenic relative to the wild-type lines. This loss of buds, especially at the earliest stages that we could see, suggests early bud abortion. And what it may reflect is the effect of ethylene inhibiting stamen development early. Now the stamens themselves are the ones producing ethylene. They would perceive it immediately, and you would have the potential then to inhibit that stamen development prior to the initiation of carpel development, at least in some of those cases, that would lead to cause asexual buds that might then be subject to abortion. And we're currently doing the microscopy to verify this view. If we express in the carpel primordia, we did not see an effect on sex expression. So the ethylene production experiments provide further support for a key role of stamen primordia in sex determination. The increased endogenous ethylene production caused an increase in carpel-bearing buds and a reduction or loss of male-only phase. And that we suggest that the increased ethylene production by the stamen primordia may lead to a loss of those initial stamens, further implicating the importance of crosstalk between the developing sex organs to assure that you have one or the other sex organ present. So we also wanted to ask, can we use any of this information to either get increased carpel-bearing bud formation or earlier carpel-bearing bud formation? Maybe we could shorten the growing season or get melons on the market sooner. So to do this, we needed to do field trials. And first we looked at 35S ACS plants, and these plants, as consistent with what we observed in the greenhouse, had earlier appearance of first bisexual flowers, increase in the number of total bisexual flowers on the main stem, and earlier appearance of first fruit by approximately 10 nodes on the main stem. If we looked at ripening, if we took a harvest date such as this example, we saw that there was earlier ripening occurring in these fruits, such that at this time point, the only lines that had either overripe or ripe fruits were the two transgenic lines. Now, this might be due to earlier fruit set, but of course it also could be due to constitutive expression of ethylene, which can influence ripening as well, and we cannot separate those effects in this experiment. We also looked at the CRC ETR1-1 melons, and again we saw earlier occurrence of the first carpel-bearing flower on the main stem, increased number of flowers, and as we had seen in the greenhouse, we also saw conversion of approximately 50% of the bisexual flowers to become female. We also saw an increase in initial fruit set on the plant by approximately 63% relative to the non-transgenic lines, but that decreased as we went through the season, likely reflecting increased abortion of these young fruit. At the end, when we got to harvest-sized fruit, there was only about a 25% increase in the transgenic lines relative to wild type. If we looked at harvestable yield per plot, it was essentially constant across all of the lines. And what we see is that that increased fruit number was counterbalanced by an approximately 25% decrease in the average fruit weight for these melons. With respect to ripening, the CRC ETR lines showed mixed phenotypes. One showed increased percent of fruit at full slip, so increased ripening, and that would be consistent with earlier fruit set and possible effect of ethylene. But one line, on the other, appeared to have a delay in ripening and did not turn orange, and its fruit did not reach full slip. And it appeared to did not undergo, this M15 line did not show a normal ripening progression. So if we're looking at our wild type melon, we're going from green to turning to ripe and then eventually overripe, where you see degradation of the flesh and separation of seeds. On the other hand, if we look at many of our CRC M15 fruit, they remain green on the outside, they do not upsize from the peduncle, yet when you open them up, there is degradation of the flesh and detachment of the seeds. If we look at internal fruit ethylene, in that one line that did show the accelerated trend as we would have expected, the M5 fruits, their ethylene production for green and ripe fruits was as you would expect, comparable to wild type. But those from the M15 line, where we had the external green but internal ripening phenomenon, we see that the green fruits that on the outside appear unripe were actually having ethylene levels comparable to ripe fruit. So can we use this knowledge to increase carpel-bearing flower production or increased or earlier fruit production? Well, both lines caused increased carpel-bearing flower production and fruit set. However, the increased fruit set was compensated for by reduced fruit size, did not result in increased yield, and the effects of ripening and potential earliness were variable among the lines and may reflect expression levels and patterns. The other area of interest is fruit development, especially early fruit growth and expansion. And we know that cucurbit fruits show extreme morphological variation, have tremendous capacity for rapid growth. There are record-setting pumpkins that are 3/4 of a ton for an individual fruit. Watermelons have been recorded to have peak growth of 0.5 kilogram per day in field conditions. And cucumbers, maybe they're not as exciting, but they can still be highly variable. So if we look at this immense diversity in cucurbit fruit shape and size, it implies that there are either novel genes or gene combinations or gene regulation capable of controlling fruit growth and development. So we're taking two approaches to study early cucumber fruit growth. The first is a Qtl-based analysis in collaboration with colleagues in Israel, Amir Sherman and Ron Ophir, and Wisconsin, Yikun Wang, as well as transcriptomic analysis of early fruit development. So fruit development typically includes 4 phases: initial development of the ovary, then following pollination there would be cell division, cell expansion, and then maturation and ripening. Cucumber fruits exhibit a typical sigmoidal pattern of fruit growth, with cell division generally occurring in about the first 5 days post-pollination, then an exponential increase in fruit size from about 4 to 12 days post-pollination, and then a period of maturation and ripening. But cucumbers are harvested as immature, typically at about 8 to 12 days post-pollination. Unlike if we were looking at melons or tomatoes, we would be out at this mature and ripening stage. The increase in fruit size is largely driven by an increase in cell size. Here we see an approximately 20-fold increase in mesocarp cell size from 4 to 12 days post-pollination. And that increase in cell size, the cells become highly vacuolated, and we see a decrease in cell wall thickness. We also see an increase in cell size in the exocarp cells, but in that case, it also includes an increase in cell wall and middle lamella thickness. So to perform the Qtl analysis, this 45,000 point SNP array spanning the cucumber genome is currently being developed by Ron Ophir and Amir Sherman to allow for Qtl mapping. And this is being done based on the availability of 2 sequenced cucumber lines with dramatic differences in fruit size and shape. The first is Chinese long 9930. This was the first published cucumber genome sequence by Huang et al. From the Beijing Genomics Institute. And the second was sequenced by Yikun Wang et al. In collaboration with Roche 454. This was a gynoecious pickling cucumber line. Yikun Wang his group also made F7 rill populations, crosses derived from GY14, the pickling type by the Chinese long. So as a starting point to identify separable Qtl that can contribute to these differences in fruit size and shape, we sought to identify growth parameters that differ between the 2 cultivars. So factors that we're looking at include ovule number, cell number at anthesis, increase in cell number post-anthesis, period of cell division, period of cell expansion, cell size, cell shape, and preferred plane of cell division. We can see that differences in ovary size and shape are apparent as early between the Chinese long and the pickling type as 7 days pre-anthesis, indicating the importance of early development factors influencing ovary shape, and in this case, ovary length. Here, there was a significant difference in ovary length at 7 days pre-anthesis, and that difference continued to occur until we hit anthesis, where the Chinese long were much longer than the pickling type. Both of the cucumber fruits exhibited a typical sigmoidal growth pattern, and as we can expect, we see an increase in cell number preceding the increase in fruit and cell size. If we look at length and diameter in comparison to each other among segregating F2 progeny, we see that they are not correlated, indicating that the factors driving increase in fruit length are separable from those driving fruit diameter. There was also a difference in ovule number, with the Chinese long approximately twice as many as the pickling type, and F1 falling almost immediately in between. If we look for a correlation between ovule number and fruit length, we see that that is correlated, but there's no correlation with fruit diameter, indicating that ovule number is a strong determinant of ovary length, but not diameter. If we look at cell number in longitudinal section, we see that at anthesis, the Chinese long already had a greater number of cells, and post-anthesis, there continues to be an increase in cell number for a longer period. Cell division is occurring longer than in the pickling type. Cell size, however, is essentially equivalent between the two. In contrast, if we look at ovary diameter, just like we saw with ovary length, the Chinese long at 7 days pre-anthesis was bigger than the pickling type. But as we approached anthesis, instead of that difference getting bigger, that difference actually disappeared. And post-pollination, we see that the increase in diameter was much faster for the pickling type than the Chinese long. If we look at increase in cell number, we see that there was a more rapid and a longer period of cell division occurring in cross-section for the pickling type than Chinese long, leading to a threefold greater cell number in the pickling type in diameter. The two lines also exhibited differences in cell shape. GY14 cell size was pretty much equivalent whether you were looking at longitudinal or cross sections, whereas there was a difference in the dimensions for the Chinese long. So collectively, these results indicate that there are several factors pre- and post-anthesis influencing fruit size and shape: the number of ovules, the number of cells in longitudinal section or cross section, the length of period of cell division and either fruit elongation or fruit width expansion post-pollination, and cell shape. So these observations suggest that there are distinct traits that we can now analyze in the real population for future Qtl analysis. And finally, I'd like to talk about our work studying transcriptomic analysis of early fruit development. And for this, a set of 3 fruit growth analysis experiments was performed in the greenhouse. We wanted to be able to monitor changes in gene expression and relate those to obvious stages in fruit development. One of the things is that typically people look at that initial stage as not very interesting. You know, the fruit's set, and then they kind of grow, and then they do the exciting stuff at the end. If they're going to ripen, they make flavors and aromas and colors. But when we started to look carefully at what's happening during that early stage of growth in cucumbers, there actually is a lot happening. There's a highly reproducible progression of changes and features, including a loss of ridges, the formation and then subsidence of warts, the development and then abscission of spines, changes in wax coverage and chlorophyll content and patterns in placenta and seed development. Another interesting feature during early cucumber fruit development is important with respect to infection by the important oomycete pathogen Phytophtora capcisi. From some work that we had been done to try to screen for cucumber fruits that were resistant to this pathogen for breeding purposes, at that time we were not successful in identifying a source of resistance for breeding. However, we did discover that there was a developmental progression in resistance, or an age-related resistance to this disease, such that young fruit are very highly susceptible and they become resistant as they develop. And this transition from susceptible to resistant was directly correlated with that transition away from the period of rapid fruit elongation. So collectively, these observations show that there's a lot of changes occurring during early fruit development, and they provide a physiological context from which to examine changes in gene expression. So to perform the transcriptomic analysis, fruits were sampled at 0, 4, 8, 12, and 16 days post-pollination. This allowed us to cover that exponential period of growth and also include the time at which cucumber fruits would typically be harvested. RNA and cDNA libraries were prepared from samples that included pericarp and placenta, but did not include seeds. 454 pyrosequencing provided approximately 1.1 million ESTs, or approximately 200,000 clean reads per time point. And approximately 82% of those could be assembled into a set of about 28,000 contigs. The mean contig length was 834, and the mean number of reads per contig was 67. Now, one of the first things we wanted to do was look at the relationship between the number of reads and our ability to assemble contigs. Of course, you would imagine as you have more reads, you can put together a longer contig. But at some point, you're going to reach the full transcript length, and it's not going to get any longer. So if we look at that, we see a steady increase in the average contig length as our number of reads increases, but it then leveled off at about 1,400 base pairs. That's a typical transcript length. That's what we would expect. And that occurred at approximately 30 ESTs per contig. We also asked what was the relationship between the number of reads and our ability to identify putative homologs in the databases. And we could see with Arabidopsis that it also steadily increased and then leveled off at about 30 reads per contig. And so we then used 30 ESTs per contig as a baseline for our subsequent bioinformatic analysis. Now at the point where we leveled off, approximately 90% of our contigs had putative homologs in Arabidopsis. And if we look at the putative functional assignments of those genes, we see that they fall into a distribution pretty similar to what you would see in Arabidopsis, within about twofold range for the categories in Arabidopsis, suggesting good representation of the genome. But there were 10% that did not have putative homologs in Arabidopsis. Now approximately 5%, approximately half of those, did have putative homologs in other species such as grape and poplar, for which there are extensive gene bank entries. But the final 5% either did not have identified homologs in the current database, or they only had homologs in other cucurbit species, suggesting that these may be unique to cucumber or other cucurbits relative to the kinds of plant species that have been sequenced to date. If we then looked at the distribution of these transcripts that did not have putative homologs, we see that that was not equivalent at the different time points. So there were approximately twice as many that did not have putative homologs in Arabidopsis at 8, 12, and 16 days post-pollination as at 0 and 4 days, suggesting that that post-fruit set, that exponential growth phase, may include more cucurbit novel genes. If we look at those transcripts represented by greater than 30 ESTs, but that had homologs only identified in cucurbit species, that included about 18 that had putative functional assignments, and of those, 8 were related to phloem, such as phloem filament protein, phloem lectin, etc. And this is consistent with the unique phloem structure in cucurbits relative to other species. We then wanted to look at the relationship between the genes expressed at the different ages using principal component analysis. And this put the different ages into 3 groups: 0 and 4 days post-pollination, 8 days post-pollination, and 12 and 16 days post-pollination. Can then superimpose that on the growth curve, and we see that 0 and 4 is that early fruit set, cell division phase, 8, peak exponential growth, 12 and 16, late or post-exponential growth. We next wanted to be able to do comparisons between gene expression at the different ages, and this is based on the assumption that the number of reads that we're seeing is actually reflective of the transcript level. And we were comforted in 2 ways with respect to that question. One was that if we looked at samples from 8 days post-pollination to sequencing runs, we see a very good correlation between those, suggesting that those that have low-level expression indeed have low expression, those that have high expression. And then we went on to select a subset of approximately 20 genes that had differing levels of expression at each of the time points. And for those, we did Qrt Pcr analysis. We saw good correspondence between the 454 data and the Qrt Pcr data. And that Qrt Pcr analysis was actually performed on 2 sets of experiments that had been done in the greenhouse at different times. So we saw good correspondence between the 2 sets of Qrt, indicating good biological conformation or reproducibility as well. We then looked at which of those constructs, which of the transcripts were most highly represented specifically at one age, not ones that are highly represented all the way across, but are there ones that are uniquely high? What's blasting at 0 or 4 or at 8? So if we look at 0 and 4, we see that those that are blasting include histones and ribosome-associated proteins. That makes sense. These cells are undergoing cell division. They need to have DNA replication. They need to have lots of protein synthesis. Histones pretty much disappear by the time you get to 8 days. There's a little carryover on the ribosomal. But what was most striking, however, is that we look at 8 days post-pollination, we see that more than half, 52% of those genes, either had unknown function or either did not have homologs in Arabidopsis, or if they did, we don't know what they do. So this large proportion of contigs without homologs or with unknown function may reflect either cucurbit unique genes or less extensive characterization of the exponential growth phase. If we then look at that group that was very highly expressed at 8 days post-pollination, and we compare that to predicted Arabidopsis homologs, what we see is very different from that earlier slide I just showed you. This is where everything else fell, within a twofold range. But here we see that there are some groups that are very highly overrepresented. And these were typically associated with cell structure. So for example, we see very high level of expression of aquaporins that would be necessary for water uptake to drive cell expansion. Other examples would be the phloem-related proteins, which would be necessary for import of photoassimilate, as well as some of the structural proteins such as actins or alpha-tubulins. Some of the other categories that were highly expressed were genes associated with cell walls. Again, you would expect that for growth, as well as several hormone-associated genes, including a key enzyme in brassinosteroid biosynthesis. And brassinosteroids have been shown to be critical for cucumber fruit set in early development, and they can induce cucumber fruit growth. Another category that was very highly expressed was latex-associated proteins and lipid-associated proteins. Well, that was looking at things that are just blasting, what's really highly expressed at each of these time points. But we're also interested in genes that may not be expressed that strongly, but still show a very specific time point of expression. So in order to get a look at that, what we did was we said, let's normalize according to the proportion of expression. So if a gene was expressed at the same level throughout this period of development, there are 5 time points, and you'd see 20% of expression at each of those ages. So now, for example, let's look at 8 days post-pollination. Let's take all of the genes and say what percentage of their transcripts occurred at 8 days. And what we see is a very strong curve with a peak almost exactly at 20%, indicating that the majority of genes had 20% of their expression at that time point. However, we can also see that there's a long tail. Similarly, if we look at 0 plus 4, now you've got 2 time points together, you'd expect 40%, or 12 plus 16, and again, they fell at exactly 40%. So then we asked, what's happening, though, in these tails? The top 2.5% of genes, can we identify transcripts that are specifically enriched? And so what we get here is 3 non-overlapping sets of transcripts, 0 and 4, 8, or 12 and 16. And not only are they non-overlapping with each other, but in large part, it was a new set of genes because earlier we were looking at the most highly expressed. They had to be at least 0.1%. On average, they would have been 1,700 to 2,000 reads per transcript. Now we're looking at ones that are on average 100 to 200 reads per transcript. When we perform functional analysis, we see different kinds of genes showing enhancement at the different age groups. So, for example, at 0 and 4 days, we see an increase, as we saw earlier, with cell organogenesis, biogenesis, DNA and RNA metabolism. And here's an example here. What we're looking at is several putative cell division-related genes, cyclin and cyclin-dependent kinases. We see very high expression at 0 and 4, and then essentially no expression after that. Another example of something that was uniquely expressed at a specific time point are several lipid-associated proteins, GDF-SF motif lipases, lipid transfer proteins, and this actually corresponds with the time period in which we're seeing both increase in cuticle thickness and change in cuticle structure. If we looked at those that are more enriched at 12 and 16 days post-pollination, we say that these had the highest representation of abiotic and biotic stress-related genes, including a variety of heat shock, redox, ethylene-related transcripts, suggesting a change in emphasis from growth and defense, from growth to defense. So in conclusion, the contig set suggests broad representation of the cucumber transcriptome. Approximately 5% of the highly expressed transcripts did not have a match or only matched cucurbit clones, suggesting that there may be genes unique to cucurbits. Principal component analysis placed the ages in 3 groups. 0 and 4 days post-pollination characterized by cell division, and transcripts most highly expressed included genes associated with cell division, organogenesis, and biogenesis. At 8 days post-pollination, we were at peak exponential growth, highly enriched for transcripts associated with cell structure, growth, lipid metabolism, and defense. And also very highly expressed genes that did not have homologs or unknown function. And finally, at 12 and 16 days post-pollination, the late or post-exponential growth exhibited increased expression of stress-related and environmental response in extracellular associated genes. So collectively, these patterns of expression are consistent with a profile of early, rapid growth followed by an emphasis on defense. And with that, I want to make sure to acknowledge the people who actually do the work. Graduate student and postdoc, Kauriandi and Murad Bicoli worked on fruit development, sex expression, Katerina Papadopoulou, Holly Little, and Jessica Taft. And the great majority of our melon transformation is done by our technician, Sue Hammer, as well as thank you to the agencies that have supported the work. Thank you.
Questions?
Yeah.
I was curious in the end, you didn't identify any transcription factors in there.
Oh, actually, there are lots of transcription factors in there. They obviously don't show up in the most highly expressed category, but we've got lots and lots of transcription factors.
There's so many intricate developmental things going on.
Yeah. We do have lots. So, but they're not, we were at this point focusing on the more highly expressed kinds of genes. Judy.
Going back to your sex expression work, the ACS gene, has anyone looked at diversity within that gene across the germplasm to see if there's evidence for selection for that gene? Because I know in maize, some of the sex determination pathways are under selection, they look like they development. And I was wondering if you found something similar.
So not so much, well, I can't, you're talking about the enzymatic activity or expression or?
Nucleotide diversity. Just raw nucleotide
just
diversity.
raw nucleotide diversity. What has been done so far, and not by us. So for example, there was a French group that cloned the different ACS genes, and the one from melon, what they had done was looking at different sex types and then pulling out extremes. So for example, the ACS7 gene in a plant that's capable of producing female flowers is functional. But the ACS7 gene in this one we work with, which is bisexual, it still produces that gene. We can detect with QRT, we can detect expression of that gene, but it's a non-functional enzyme. It has a mutation in an active site. So.
This has been a production of Cornell University, on the web at cornell.edu.